Well, I'm a research microbiologist. I work in the Division of Healthcare Quality Promotion and a laboratory called the Biofilm Laboratory, a laboratory that we started about 12 years ago. I'm actually trained in microbiology and more on the environmental side. I consider myself an environmental microbiologist and a microbial ecologist, really working in the arena of public health and in clinical microbiology. One of the first things that I've really focused on here at CDC has been to write review papers and book chapters, a number of those, to try to get information out to the infectious disease community on the importance of biofilms and infection. That's really the first contribution I think our lab has made. I've seen our work focus in three areas, broad areas to do with biofilms and public health. One has to do with biofilms in potable water systems, the role of biofilms in those systems in terms of the survival of pathogenic organisms, for example, in biofilms. In other areas had to do with antimicrobial resistance, understanding the role of biofilms in the horizontal transfer of resistance genes in biofilms and the dissemination of resistant organisms. And the third area, of course, has to do with biofilms on indwelling medical devices. One of the big changes for me has been a better understanding of biofilms as microbial communities. We need to understand that it's more than strictly just an organism attached to a surface. The organisms in biofilms interact with one another. And that's important in terms of their ability to attach to a surface, to grow and develop a biofilm structure, and to be dispersed into the environment or into the patient's bloodstream, for example, and how different organisms interact in terms of things like quorum sensing. We've seen the development of new methods for actually examining biofilms directly on surfaces. So we can look, for example, using molecular probes, fluorescent in situ hybridization probes, or various kinds of fluorescent dyes, at methods that will allow us to examine the interaction of cells in a biofilm or examine the effect of a disinfectant or an antimicrobial agent on the biofilm cells. I think we've seen a real development of the use of non-culture-based methods for determining the extent and the diversity of microbial communities, and also, of course, the role of biofilms in things like chronic wounds and understanding that a little bit better. I might use, for an example, a chronic infection would be something that's ongoing, that's sort of recalcitrant to antimicrobial agents. For example, biofilms in osteomyelitis or biofilms on a prosthetic joint or an intravascular catheter. An acute infection might be something that's more easily treated, something that's more easily detected. I think if we understand Koch's postulates, I think at the time, and in infectious disease microbiology, I think the Koch's postulate approach is very valuable for understanding the disease process, the ability that we can take an organism, isolate it, put it back into an animal, and produce symptoms of a disease. But I think probably it's quite true, I think it's well supported that there are certain diseases that have a biofilm component that may not adhere exactly to that process, in the sense that we might have multiple organisms involved in a disease process. And there could be other non-pathogenic organisms that could play a role in the disease process. The whole concept of opportunistic pathogens, organisms like Pseudomonas aeruginosa, which may not be frank pathogens, but can be very important under certain conditions in the disease process. So I think that's an important consideration. One of the things that's really interesting to me is not so much that we find an organism in a biofilm, and even that organism, for example, on a medical device, is related using molecular methods to the bloodstream isolate. But what's really interesting to me is virulence. What are the factors involved in biofilm formation that result in the expression of virulence by bacteria? I think that's a really important question. We really don't have an answer for very well. There's certain groups working in that area. It's something that's very important for us to understand, and so that we can understand the role of biofilms in infection. In terms of treatment, we know that organisms in a biofilm are very recalcitrant to antimicrobial agents. They're very tolerant. And so that tells us that the standard protocol that we use for treating biofilm-associated infections is not going to work very well. The literature bears that out. Systemic drug treatments, for example, are not very effective against device-associated infections. So that's one of the areas important. We need to know, of course, biofilms are very tolerant to the immune system. the immune system is not very effective at eradicating biofilms. Organisms in a biofilm tend to become more tolerant and more difficult to treat as they age, as the biofilm ages. And so we need to have a different approach in terms of treatment for biofilms than we would for normally processes that occur under more, where the organisms are planktonic, which means suspended and not associated with a biofilm. One of the things that happens to cells in a biofilm is that their growth rates tend to change. We've observed this. We've observed that with certain bacteria, for example, they grow much more slowly in a biofilm. And that's very important for certain kinds of drugs. For example, the glycopeptides or the penicillin type drugs, which are based upon the growth of the organisms. The organisms are growing much more slowly, therefore the drugs are not going to be nearly as effective at treating them. So that's one of the proposed mechanisms, growth rate. Another has to do with the fact that biofilms, as they become established, develop different chemical and physical gradients. For example, gradients of oxygen that might tend to protect the cells from the antimicrobial challenge. Another has to do with the structure, the structural matrix, the extracellular matrix, the slime, if you will, that's formed in a biofilm structure that will in fact protect organisms from diffusion of the drugs. And then there's even some work pertaining to the survival of certain persister type cells, cells that are just naturally more resistant to the agent and being selected for. So there could be a number of mechanisms in terms of their tolerance. I will state that I'm using the word tolerance because I think tolerance is different from resistance. When I think of resistance, I think of a genetically based mechanism for the ability of the organisms to resist the drug. And biofilms can be important there as well, because through genetic exchange, through conjugation, for example, cells can, in a biofilm, have been shown to have an enhanced ability to exchange resistance factors one to another. I think that I'd speak, for example, of a resistance to a specific drug. In that case, there could be genetic exchange. So in that case, we could see either through conjugation or transformation where the cells take up the DNA containing and encoding the resistance factors in a biofilm. And that's been demonstrated in the laboratory. That's one mechanism. Another is the fact that the cells are just naturally tolerant to the various antimicrobial agents in the biofilm community. There is some literature suggesting, too, that organisms can have a protective effect one to another. So, for example, in the laboratory with polymicrobic biofilms, we've seen evidence that one organism might provide a protective effect for another organism in the biofilm. So that could be sort of a third mechanism for resistance. I would define a biofilm as a microbial community of organisms associated with a surface with one another or with an interface that expresses genes that result in a different phenotype with respect to growth rate and tolerance to antimicrobial agents, and tends to produce an extracellular matrix or slime surrounding the biofilm that resulting in a structure of some kind. And I think we do need new approaches. I think the old approaches aren't very effective. One of the things we could do right now, I think, is have a recognition in the healthcare community that biofilms happen, that they're important, that they don't respond the same way that what we call planktonic or non-biofilm cells will respond. So therefore, the first thing might be in the way that we test and evaluate different treatments against organisms. I think it would be helpful to provide new testing protocols that could be standardized, that could be accepted, and could be utilized by healthcare practitioners for treating organisms in a biofilm. That could be a first step.